So why does this turn into this in the fall? I live in Boston, which means that about this time each year, the leaves on the trees start to turn their colors. Leaves get their color from 3 main compounds. The first is chlorophyll, which might sound familiar. Chlorophyll is involved in photosynthesis, which is the process by which plants turn CO2 and water into sugar and oxygen by harnessing the energy from the sun. Chlorophyll gives most plants their distinctive green color. The second group of compounds are the carotenoids, which sounds a little bit like carrot, which makes sense because carotenoids give leaves their yellow and orange hues. Carotenoids are present with chlorophyll in organelles called chloroplasts year-round, and these chloroplasts are just the organelles where photosynthesis occurs. Finally, there's a group of compounds known as anthocyanins, and anthocyanins give plants sort of reddish hues, and they're often found in things like cranberries and blueberries and lots of dark red leaves. These anthocyanins are actually produced in the fall when chlorophyll starts to break down, but I'm getting ahead of myself. Now, for most of the year, chlorophyll is so bright green that it blocks out some of the yellow and orange hues coming from the carotenoids. For most of the growing season, chlorophyll is constantly being broken down and reproduced. But as it starts to get colder out, the cool night air closes off the veins of the leaves, and it gets harder for water and nutrients to move into the chloroplasts and rebuild chlorophyll. So what this means is that the chlorophyll is being broken down, but it's not being reproduced, so the green color of the leaves starts to disappear. This means that the yellows and the oranges of the carotenoids can start to show through. But that's not all. Anthocyanins, by contrast, are not present year-round in the leaves. Instead, they are actively produced in the fall, right before the leaves drop off the trees. So why would a plant suddenly want to start producing a red pigment in a leaf it's about to drop off anyways? Scientists haven't reached a definitive conclusion yet, but they do have some pretty good ideas. One is that the red pigments act as a sort of direct sunscreen as the canopy starts to drop leaves. The leaves that remain are exposed to more light, and those leaves are losing the sunlight absorbing powers of their chlorophyll. Another cool idea is that the red pigment might actually trick migrating insects into thinking that the tree is unhealthy and discouraging them from laying their eggs on those trees. So all of this leads us to some pretty cool science that you can do at home to visualize the pigments found in leaves where you live. So all you need for this experiment is isopropyl alcohol, some strips of filter paper, I used coffee filters, a couple glasses, scissors, and some leaves. And if you want to be super cool, pick a few different kinds of leaves so that you can compare the compounds you find within them.
In this video, I'll show you the protocol that's one of the core practicals in IB biology, illustrating how you can separate leaf pigments by a process known as chromatography. In this example, I'm going to use spinach leaves. So the first step is for you to obtain your plant that contains the pigments you wish to separate. Break up one of the leaves into little pieces and place it in a mortar. Add a few drops of acetone and a little bit of sand, as this helps with grinding up the leaves. Use the pestle and mortar to grind up the leaves and to release the pigments. If it helps here, you can add a few more drops of acetone, but ultimately the objective is to obtain a concentrated pigment in a liquid at the bottom of the mortar. After you've done this, then you need to prepare your chromatography paper. Now, this does actually work best with thin layer chromatography paper, but you can also use regular chromatography paper. To prepare, you're going to obtain a pencil, and it is important that it's a pencil, not a pen, because a pen contains other pigments. Use the pencil to draw a line on the chromatography paper that's about 2 centimeters up from the bottom of the paper. You can see that here. At this point, you're going to place a spot of concentrated pigment on the line. Now, this can be done in several different ways. Here, I'm using a very small, narrow plastic pipette. It can also be done with a capillary tube, and sometimes people do it with a very, very fine paintbrush. But the most important part is to make that spot as concentrated as possible. So in between spotting, you can dry it with a hairdryer to make it more concentrated and then spot on top. And you're going to keep doing this until you have a very, very dark green concentrated spot of pigment on your line. The next step is to prepare your running solvent. So you're going to take the container in which you will place your chromatogram, and you're going to pour your running solvent into the container, ensuring that it does not go above where the pencil line is at on your paper. So in our case, that would be not above 2 centimeters from the bottom of the container. Now, there are a variety of different running solvents that people tend to use. One of the most frequent is 9 parts petroleum ether to 1 part acetone. but it is possible that your teacher might give you a different running solvent to use. In any case, you would perform the same actions here, but just with the solvent that you were provided with. Once you've prepared your container, you place your chromatography paper gently into it and allow the solvent to begin to move up the paper, carrying the different pigments with it. Once you've marked where the solvent went to, then you need to measure the distance it travelled from the point of origin, which is the original pencil line that you drew on the chromatography paper. This is known as the distance travelled or moved by the solvent front. Next, you want to mark the front edge of each pigment, so that's each colour, and you want to measure how far each pigment or each colour moved from the point of origin, the original pencil line that you drew. In order to identify the pigment, you first need to calculate the RF value for each pigment that you found on your chromatogram. To do this, for each one, you want to take the distance moved by the pigment, presumably in millimeters, and divide this by the distance moved by the solvent front, also in millimeters. Once you've calculated the RF values for each pigment that you observe on your chromatogram, you can compare it against known published RF values in order to identify the pigments on your chromatogram and therefore the pigments in your leaf. For example, the published RF value for carotene tends to be between 0.89 and 0.98. So if you obtain a number within this range, and it tends to be a yellow-orange colour, it's likely to be the pigment carotene. You can see it here at the top of the chromatogram, very close to the solvent front. The published RF values are easy to research and look up, and there are many tables available for you to compare against and identify your pigments. But for example, this website also offers additional resources, including the written protocol, to help support you when carrying out this practical in the lab.